thanks everyone for coming. My training was as an ecologist, and so I have not been trained as a taxonomist or as a molecular biologist. So it took me a while to get on board and to realise, exactly how profound some of these changes have been. I have done my time in the lab over the last few years, extracting DNA, running PCAs, trying to interpret chromatograms, and exploring other ways to look at species boundaries in corals. And so, I think this is one of the most important areas of reef research at the moment. It's a big problem, and it's going to take a lot of effort to end up with a complete phylogeny of corals. But so today, I will outline the molecular revolution. I'll let you know how it developed, and then I'm going to attempt to summarise some of the major changes. So these are the changes that we all need to be worried about, how species have been shifting amongst families and amongst genera. And then I'm going to present a case study, some work I've been doing with Josh Maiden in the Solitary Islands, and again, I just want to show, using molecular approaches, I hope we've avoided a lot of the mistakes of the past. And in terms of biodiversity research on corals. And then finally, I'm just going to discuss some of the implications and some of the work that I think potentially needs to be redone, and some of the opportunities. Because as I said in the abstract, I think this is an exciting time to be doing this sort of research. So, coral taxonomy. It's based, until recently, almost entirely on morphology. It's no accident. I think most of the early work on coral taxonomy was done by paleontologists, and so there's a limit to the amount of information you can get from a fossil. But people have known for a long, long time that there are problems with morphology as a guide to species boundaries. In particular, there's extensive morphological variation within species, and it's meant that the boundaries between species have often been fairly arbitrary. And so this is a very famous series of photographs of corals that was published in Charlie Baron and Michel Pichon's monograph back in 1976. And they used this diagram to illustrate phenotypic variation in what they believed was a single species. So when Charlie and Michel began working on the Postlepora, this is Postlepora damcornis, this was at the time believed to be about 3 to 4 different species. They reinterpreted it as changing in environment. So the corallum up here is from a reef crest, high energy. Down here they interpreted it as the morphology this species took on in more less energetic environments. And ironically, this is probably now the poster for the molecular revolution, because we've come full circle. And now this is again realised that this is actually 3 or 4 different species. So what goes around comes around. Again, so we've always known there were problems with morphology as a guide to phylogeny, but it wasn't until a paper back in 1996 by Sandra Romano and Steve Palmumbi that people's eyes were really open to the scale of the problem. And what they found was that they found 2 major divisions in the this is a phylogeny, the phylogeny they published based on the 16S ribosomal gene of the Scleractinia. And the main finding was these 2 big clades. They date back about 250 million years to the division there. And so they've put their molecular clade together, and they've mapped onto it the old morphological ideas of the phylogeny. So what you can see is that species or groups that people thought were very closely related, so the Postleporas and the Aquaporas, believed to be very closely related and were put in the same suborder, actually on either side of this big division in the phylogeny. And again, so their nearest common ancestor was 240 million years ago, whereas people thought these were sister taxa. Other examples, so here are some agaricids, and the fungids, people thought they were very closely related. Again, their nearest ancestor, 240 million years ago. The next, I think, major advance was some work by Alex Kerr. Alex did work here at James Cook University. And again, very similar findings. Alex put together a super tree. So he grabbed all the phylogenies he could find, morphological and molecular, threw them into an algorithm that gives you a phylogeny. And again, so Alex recovered these two major clades that Romano and Palmumbi had discovered. And again, I think the major finding here was that nearly all of the families were spread within this phylogeny. Very few of them were monophyletic. And so here is one example, the Scleractinian. It actually comes out in 4 different clades at this level. And again, on either side of this big division in the Scleractinian family tree. Similarly, the Caryophyllidae, again, I think split into 5 big clades. So this was the first indication that there were major problems and that morphology, that it guided all these decisions about phylogenetic relationships, was just not particularly reliable. Now, Alex's paper doesn't get nearly the credibility, the respect it deserves, so I just thought I'd plug his other career as a guitarist in the band. And there's a very angelic-looking Morgan Cratchit there too. So the paper that does get all the credit is this next one by Phylogeny, which deservedly so. This paper, again, reconstructed a phylogeny based entirely on the genes, and they found that there were approximately 20 molecular clades or families, and none of them were monophyletic, or very few of them. Anyway, so that's the revolution. And you know, it was at this point that people knew that there was something very seriously wrong with our taxonomy. Again, this is another example of, just to demonstrate how badly a phylogeny constructed by morphology and one constructed by molecules agree, the huge disagreement between these different approaches. So this is a paper by Roberto Aragoni. Here he's drawn his molecular phylogeny. That's the tree. And again, you can see that the various genera of the Dendrophylliidae are scattered throughout this tree. So these were the morphological-based genera. They're scattered throughout the tree. But the other thing they did was look at some of the morphological characters that Steve Cairns had used to construct his phylogeny. And again, they showed that none of them were uniquely identified with particular clades. So again, some fairly convincing evidence that all of these morphological features, and some of them micromorphology as well, were not particularly reliable. And this is what they concluded, that none of the micromorphological or microstructural characters used by Cairns are uniquely present in any of the molecular clades. So, about 8 or 10 years ago, people realised that there was a huge issue. And most of the work since then has been in trying to clean up the mess. I just put this one up here to show you what was probably one of the biggest mistakes in scientific history. It's poor old Wells's tree of the Scleractinia, based largely on the fossil evidence. And I think practically nothing there is right. None of these families were monophyletic. None of these evolutionary relationships were correct. So, this is literally the foundation of coral taxonomy systematics fell apart. So, yes, the next stage was to try and clean up the mess. And so Nancy Knowlton, working with colleagues, gave a group of these clades, some of these old morphological families, the Favids, the Merulines, Pectinus, Trachyphyllidae. She called this clade the Big Messidae. And so this is a paper that I'm happy to be associated with because it's got such a good title. And yeah, look, and that's been the work since then. And there's been a lot of work, and a dozen papers or so. Again, not many of them very high profile, so not many people will come across them. I've got a list of them, I've got PDFs of them all. So I will do what I can to distribute them if anyone wants them. But in the next few slides, I'm going to summarise some of the changes again. So just to let you know how fundamental some of these changes have been. So this is a graph of the former family Fabidae. And so here we've got the genus, as listed in around 2000, and then the molecular genus, and finally its molecular family. And so, no need to look at the details, I can give them to you later, but most genera are synonymised or split. Very few of these genera are monophyletic. So, as you can see, Diploria has been split in 2, Fabia has been split in 3, and probably the biggest changes have been to the genus Montastrea. And so, previously there were 12 or so recognised species, and most of them have been changed. So these Montastrea annuligula and curta are now recognised to be in the genus Astrea. These other Montastreas are all now placed in the genus Phylogeny, and so on. Quite fundamental changes. Again, so here is just an illustration of how some of the traditional morphological features we used are unreliable. This is a picture of Goniastrea retiformis. One of the defining features for this genus was shared walls. This is a photo of the former Fabius deligera. The defining feature for this genera was that it didn't share walls. And the molecular work has revealed that these guys are actually sister species. They're separated by less than a couple of million years. And so again, that's just a sort of graphic illustration of how these fundamental features of morphology that we use to distinguish the genera are unreliable. Again, the new family Lobophyllidea. A lot of this work is still in press. So I put this together over the last few days by looking at a few PDFs of papers that are due to be released quite soon. And again, fundamental changes. This is a new family. It's one that had to be created because because the old family Mustidae is no more, or there are a few species in it over in the Atlantic. Yes, so it includes genera from 3 different morphological families. So this is a new family that's drawn in genera from elsewhere, so from the pectinids and the phallids. And again, most of the morphological genera have been synonymised or split. And the main changes have been to Acanthastrea. The species in this genus are now spread amongst 5 genera in 2 different families. And so, so things like Symphilia is now no more. So species and genus that we've been familiar with for many years that are reasonably common on the reef crest are now gone. I mean, we need to call them something else. I suppose one of the things from one of the results of this is that most of the species have stayed the same. So while the features that define the genus and the family were not particularly informative, if you knew how to identify Acanthastrea maxima, and if you've identified that in your data set, then you're okay. You're in reasonably good shape, because we know that it was just assigned to the wrong genus, and so it's a very simple thing to change the name of that species in your database. So most data that was collected at the species level is still okay. If, however, you collected your data at the level of the genera or the family, forget it. There's nothing you can do. If you named your colony Acanthastrea, well, it could have been one of 5 different genera in 2 families. So there are big problems. There are issues at the species level too, which I will move on to. Okay, so any good news amongst all this carnage? Not much, but some of the more diverse and ecologically important genera are at least monophyletic. So it would appear so far that the Acropora and the Montipora and Prides are probably monophyletic. The Acropora almost certainly so. But how good is the species level taxonomy in these genera? Not very good. So as part of some work I've been doing in the Red Sea, I was given the unenviable task of trying to sort out the Montipora. Work I've been doing with Mike Berriman over at KAUST. there are very different ideas of the biodiversity of the region. There have been 3 major revisions. Some authors think there's 12 species. When Charlie did his work, he identified 31 species. So there are big differences in opinion. So I realised I would have to go back to the drawing board. And so in total, there have been 230 species of Montipora described in the literature. In the last major revision of the genus, Vron and Wallace reckoned that only 80 of those 230 were valid. They synonymised 90 of them, so they reckoned that 90 of these species were just the same as some of these 80. So that accounts for 170 of the species. But there's still 60 nominal species that they didn't deal with. Admittedly, their revision was on the GDR, so you might not have expected them to deal with it. But so it means there's a big mess out there still to be solved. There are 60 nominal species, and they don't appear in any of the guidebooks like they're on 2000. They're just lost. You can find them in worms, but otherwise the World Register of Marine Species. There's enormous problems that still need to be resolved in the taxonomy of these genera. The Acropora is just as bad. Okay, the way forward. It's going to be difficult. In genera where we have good molecular markers, so Acanthastrea, Montastrea, it's relatively straightforward. You can use these molecularly defined groups to go back and look for good morphological characters, which is what we need as ecologists. But in groups like the Acropora, Montipora and Parietes, where there are as yet no good markers, it's going to be really difficult. And I don't know what to do. Do I wait 10 years until they find the good markers? Or do we try and persist with more in-depth morphological analysis? I just don't know. But, that's the goal now, is to find good markers and then go back and find morphology markers that correspond to these molecular groups. Again, the other difficult thing at the moment is communicating these changes. They are scattered throughout a diverse literature and often an obscure literature. So somebody needs to do Veron 2000 again, so we've all got a readily available resource. Tom's put his hand up. So that's another major issue at the moment, is communicating these changes to ecologists. Okay, so now for a case study of our work in the Solitary Islands. The Solitary Islands is a group of islands off the coast in New South Wales, off Coffs Harbour, about 300 k's south of Brisbane. I visited there with Sebastian Smith-Roach, who was collecting for his revision of the Oscillopora. And just in one dive in the Solitary Islands, just taking photographs, I saw 3 species at least that hadn't been recorded there before. And so we wrote it up in this reef site, and we hypothesised that this was evidence of range expansion of corals due to climate change. These sorts of range expansions have been seen in the fish, so it was a reasonable hypothesis. There have been big changes in the fish assemblages in the Solitary Islands, and there has been, I think, a 2-degree change in the average winter minimums over the last 30 years. So there are definitely changes happening there, and so we hypothesised that these changes might be due to climate change, and we got some money from the New South Wales government to look into it. So the aim of the work that we did down there was to quantify changes in the coral assemblage. And so to address that question, we went down and did field surveys. We collected a lot of samples, and we did molecular analyses as well as morphological analyses of our samples. It was another ideal location because there's a good historical data. Some of the first work that Charlie Veronica did was in the Solitary Islands. So some of the first work published in Australia, and including species lists. Vicky Harriet did this work, similar work, in the 90s. So we had good data, approximately 20 years apart. And the other thing we had access to was a collection that Charlie did in the Queensland Museum. So we had actual coral specimens from the 70s to go and check. And so this is just an illustration of the changing ideas of the species richness in the solitary islands between these three studies, separated by 20 years. So Charlie listed a total of 31 species back in the 70s, Vicky 69, and we reckon there's about 68 species. And you can see that there's not a lot of overlap. Of those 95 nominal species, only 24 of them are common to each of those studies. And so if you compare our species list to Vicky's, there's about a 50% turnover in 20 years. So that's evidence of some pretty major change in the assemblage. And yeah, so we were interested in whether, was that real? So we went back to Charlie's collection for a start. And me and Francesca Benzoni went through and looked at all these specimens and reinterpreted them. And we found that it's about 21 out of the 42 or so specimens we didn't agree with the IDs, and I think some of them were clearly wrong. Some of it was unavoidable. Many of the species were not named back in the 70s. And again, Charlie's updated his lists, so they don't include a lot of these early names. But some of these could have been avoided if the molecular approach was available back in the day. And this is just an example. This is one of the species that we've been looking at. What we've found in the solitaries is that every plocoid myriad alignment that we've sampled has been of this one species, but it's incredibly variable, and you could quite easily see that people would assign it to a number of different species. So this is how Australia, formerly Montastrea curta, looks on the Great Barrier Reef. This is a typical colony in the solitary islands. This is another colony in the solitary islands. And again, this one here it's very plocoid. Here it's almost serioid. So I can easily see someone calling this dividers of deep, which I think is what happened. And this is what it looks like on Lord Howe Island. So this is just one incredibly variable molecular species. At least that's what we think now. And so, a lot of the diversity, particularly in Vicky Harriet's, was she saw a lot of what we call Dixastreas. And we're reasonably confident that they're all Montasteras, or Australia. Another example is Paragoniastrea, formerly Goniastrea australensis. Again, it's a serial. It's typically meandroid massive. This is its sort of typical shape in the solitary islands, but you could easily confuse this with a platygyra. And in fact, that's what I did when we were sampling them. I thought there was both, just based on our field surveys, I thought there was both platygyra lamellina and platygyra dedalia. And they were on our species list until I sent samples across to Dan Wayne in Singapore, and yeah, they all came back as belonging to this new clade, newly defined molecular clade, Paragoniastrea australensis. This is what it looks like on Lord Howe Island, again, radically different, really steep walls, wide valleys, and some of the species are almost entirely, some of the colonies are almost entirely serioid as well, so I think people have again confused it with Phylogeny and other things. So, without the molecular work, I think we would have repeated the mistakes that these guys did. Our estimates of the biodiversity would have been much higher than what they are. And so, based on our revisions, we revised Charlie's species list based on the museum collection, and taking into account differences I think Vicky overestimated the diversity because she saw a number of these species which we hadn't seen. The estimates of turnover go from 53% down to about 14%, which I suppose is still high. The numbers are increasing, but it's a bit less obvious that there are major changes, or that, there's no evidence for major changes in the assemblage. So just on that theme, to show you, those estimates of turnover are based on the species list. We also looked at assemblage structures, so we're running our point intercept transects to get relative abundance of all these genera. And here, this graph compares the cover of the sort of 17 most abundant genera in 1992. So this is Vicky Harriet's work, same sites, same technique, to our work in 2013. We've defined each of these 17 species, which are the dots, in terms of whether they're cosmopolitan, meaning they're abundant in both locations, or subtropical, meaning they're abundant only in high latitude areas like solitary islands. And you can see that there hasn't been a huge amount of change. Some have gone up, some have gone down. But of the ones that have gone up, 4 or 5, they are all subtropical species. So Aquapora glauca, number 3, Acanthastrea echinata, number 1, and number 10 is Micromussel orinoensis. So, and again, one of the species that seems to have declined most, ironically, is Aquapora solitariensis. So if we're seeing anything here, it's not a tropicalisation. It's getting more subtropical, which is very different to what's happening in the northern hemisphere, where reefs in high latitude, there has been a phase shift from algae to corals. So hyacinthus is marching north at 14 kilometres per year, and it's starting to take over some algal beds up in Japan, which is causing economic hardship because people actually farm the algae. So some people might think reefs on their doorstep would be nice, but not everyone. Okay, and finally I just wanted to show you, compare the structure of our solitary island coral assemblages to that on Lizard Island, so a typical Great Barrier Reef site. This is just the percent cover in each of these different families. Lizard Island in black, solitaries in white. You can see that Lizard Island is a typical GBR community dominated by Aquapora, but the solitary island is very different. It's dominated by Myriolinids, Lobophilids and Dendrophylliidae. And so again, I think the revisions that we've done down there have revealed that these assemblages are much more unique than previously recognised. In particular, there's at least 2 endemics. So the Possipora iliciae that I mentioned earlier, that Sebastian has described recently, and there's another Cyphastrea, which we're in the process of describing now. And these are almost certainly subtropical endemics, almost certain there's Pontiporas and varieties that are endemic to the region, which brings it up to close to 10% endemicity. And again, very narrow ranges. So that's the summary of the work. The turnover, I think, is much lower than suggested just by the species list. I don't think there's any evidence for an increased tropicalisation, and the assemblage is much more distinct. And that's important because solitary islands are bleaching just like practically everywhere else on the planet. And so this is a photo that Mike of Jenny Dives sent or posted to the web. And this is that endemic species, Possipora iliciae. Most of it is, as you can see, 100% bleached. So there's going to be very high mortality of this species. And if this bleaching event is really severe, we could see some of the first extinctions on the planet in corals. Okay, so finally, a few of the implications for coral reef ecology. And in particular, I just want to revisit some of the ideas we have about evolution, biogeography, that probably need to be re-examined in the light of the molecular revolution. This is a very famous photo of image of Stellium Wells, 1971. It's contours of generic richness. The changes at the level of a genus are quite profound. I think in those big clades where the changes have been, the Myriolinids, the Lobophilids, they contain about 30% of the generic richness. So, these sorts of contours probably need to be revisited. I'm not sure things will change much. I mean, they pick up a centre in the Coral Triangle and another centre in the Red Sea. Not sure. But I think it probably needs to be revisited, as do species level contours. Range size distributions. This is some data from a paper of Terry's in 2002. What it shows is the frequency distribution of range areas in corals and fishes. And corals are quite unique. They have an overabundance of species with incredibly large range sizes. But this is, again, some recent work, 2013, Alan Chen's lab, essentially. This is the molecular divisions within the species Stylophyllidea. And so again, I think it's becoming apparent that there's probably, they've identified four clades. They haven't taken any formal taxonomic decisions as yet, but it's almost certainly at least 4 species. And that pattern is going to be repeated in lots of the pandemic species that make up that end of the distribution. Hostlepora gammicornis has now been split, 4 or 5 species. Grays lavata, some of these species that occur all the way to the eastern Pacific. They're almost certainly species groups. And so, again, I think that range size distribution probably needs to be revisited. Again, more work of Terry's, where they were looking at the life history of endemic and pandemic corals. So, again, based on those distributions, they've divided the species into endemics and pandemics. And then they've asked whether there's an overabundance of species in any of these families, in either of these groups. So, for example, there's a lot more Acropora, so you would expect Acropora to make up a lot more of the endemics. And so they concluded that there really was no signal in these patterns, except in the agaricids. So these are the agaricids. There's more pandemic agaricids than you would expect, based on their abundance in the species pool. But again, none of these families, or very few of these families, are relevant. It would be easy enough to shuffle the species around into their current molecular families and re-examine this issue. So, just to finish on a positive note, these are exciting times. There is an extraordinary amount of really good information out there. The World Register of Marine Species has all of those changes that I showed you, that I summarised today. It's all there. You look up your previous molecular morphological species and it will give you the current synonym, the molecular species. And I've got it all summarised in a spreadsheet which is available at Coral Traits as well. We also have a working phylogeny which includes Rich Dan-Wakeler published in 2012. I think there's been another one since. I think 1,200 of the 1,600 odd coral species are included in that phylogeny. Obviously, there are areas, problem areas like all the species, but the deep branches are pretty solid. And so there's a lot of really interesting questions that you can ask with that phylogeny. The distributions are out there. Terry's made all his data on species level distributions. Charlie's working on his CoralGeographic. So obviously I think some of these distributions will need to be revisited. But again, there's a hell of a lot of information out there, and much is still good. And coral traits. Josh Maiden and I and lots of others have spent the last 5 or 6 years compiling coral traits, lots of different traits for corals, and again, we just recently made that data all available for free at that website. Anyway, so it is an exciting time, I think, to be a coral reef ecologist. And thank you to everyone.